I'd like to thank the organizers for inviting us this morning to give you an update on GrayBug. Our technology has allowed us to develop a product that we're moving forward with into the clinic that can allow you to dose once every 4 to 6 months for the treatment of wet AMD. It blocks both the VEGF and the PDGF pathways from a single agent, and we think this would be a significant benefit to patients because, as you know, once Vovista is approved, we will have up to potentially 20 injections per year. We can then reduce that down to 2 to 3 injections per year for the treatment of wet AMD. We are currently on track to file an IND for this indication next year, and I'll talk a little bit more detail about the plans for that program. The technology is very novel in the sense that it's not any different than what you're currently doing to treat patients today with Lucentis and Eylea. It's a simple injection of a suspension of particles, and I'll show you a little bit more about that technology in a few slides. It's very easy to administer. It has minimal side effects. We don't see the typical inflammation from these types of particle systems, and it's really, I think, a significant advance in the technology. We licensed this technology, and I'll talk a little bit more about it, from the Wilmer Eye Institute. It was created there, and it actually allows us a broad platform to pursue multiple different indications. Beyond AMD, we're also looking at glaucoma. And in addition to just the platform drug delivery technology, we've come up with novel chemical entities that allow us to do things that some other folks have never been able to do before, and that is get IOP lowering and neuroprotection from a single agent over a 6-month period of time. So we're in the very early stages of working on that. In addition, we've been fortunate enough to be involved in a collaboration with Aerie Pharmaceuticals, which just started in August, and they've been very helpful in working together with us to take their compounds and put them in our delivery system. And then finally, we've been able to work with a very experienced team. Tom mentioned that we've known each other for many years. I worked at Genentech in the 90s on Lucentis, the very first version of Lucentis, and also developed a sustained-release formulation of Neutropin Depot, and launched that product. In addition, we've been very successful in bringing on board a very experienced team. Christy Schaefer and Jerry Cagle have been involved in many ophthalmology companies and products over the years and been very helpful. And then, as I mentioned, the technology came out of the Wilmer Eye Institute. The original experiments were done by Justin Haynes, Peter Compacchiero, and Peter McDonald, who discovered that by hydrophilic coating these polymers, we could significantly reduce inflammation. And then more recently, we've had even greater advances to show that we can get these particles to aggregate upon injection and minimize any interference with the visual axis. So the technology itself involves microparticles that can be injected very easily, as I said, similar to how Eylea and Lucentis are administered today. Really minimal inflammation, no more than what you typically would see with an injection of Lucentis or Eylea. In addition, the particles aggregate together, and I'll show you a video of how this works, and form an implant. So we don't have to do an implant. We don't have to manufacture implants. These are very simple to make particles that aggregate together. And we've already identified a CMO that's made particles like this for many years for ophthalmology indications. And we've also familiar with this technology. It's been around for about 30 years to make microparticles. So it's not a new technology in terms of the manufacturing. We're in the middle of a rabbit study right now, so I'm not sharing the detailed data. We hope to share that with you at ARVO. But we have already demonstrated after 3 months post-injection, 1,000-fold the Ki of the molecule is sustained in the RPE and the choroid. So after a single injection, we're already getting very therapeutic effects out to 3 months. The study is ongoing all the way out to 6 months, so we believe this formulation has at least an every 4-month profile. We have not seen any inflammation or by histology any toxic effects on the retina or any of the other ocular tissues to date. So we'll be very excited to share the detailed data with you at ARVO. So this is how the technology works. We encapsulate, and I said microencapsulation has been around for about 30 years. It's a very straightforward procedure. We encapsulate the drug inside of our microparticles, and we then inject them into the vitreous. And if we can get the video to play, I don't know if someone can make that work for me. That would be great. So as you see, when the injection is going in, and typically we wouldn't inject straight into the visual axis, but this is so you can see it. We're injecting through the pars plana. You see the particles coming out as a continuous gel. It's in a viscous vehicle. Over time, as they go into the eye, they aggregate together, and they form a solid implant-like structure. And you can see that on the left side there with the fundus photo. That's after 7 days, but it happens pretty much immediately. And even out now to 3 months, we're seeing an intact implant structure with no free-floating individual particles in the visual axis that can be observed by fundus photography or slit lamp exams. So the other question, obviously, is does a molecule have a long-term therapeutic effect? This first study was done by Dr. Compacchiero and Dr. Haynes at the Wilmer Eye Institute. where they gave a single administration of our lead program that has, again, blocks VEGF and PDGF, and then looked at CNV laser treatment in these mice after different times. And I just give you two examples here. One where they laser treated after 4 weeks post-dose, and then one after 8 weeks post-dose. And you can see a significant inhibition of CNV area, even out to 9 weeks. Dr. Campuchiero didn't think it would last this long, so we didn't have another group of animals to continue the treatment. We're currently doing studies in rabbit CNV models all the way out to 6 months to demonstrate the relationship between the pharmacokinetic profile that we see in the ocular tissues and the inhibition of laser-induced choroidal neovascularization. And then in addition to that, the glaucoma programs, and I think you've heard a lot already this morning, so I won't belabor the point about glaucoma, but the unique twist that we have on this is we're making actually novel or new chemical entities that take the same actives that are already approved and on the market and create prodrugs that allow us to release those actives. In most of the talks you've heard about prostaglandins, we can actually do this with carbonic anhydrase inhibitors. We can combine prostaglandins with carbonic anhydrase inhibitors as a single agent, so we don't have to do combination approaches in clinical trials. And to take that one step further, we're actually developing compounds that have both IOP lowering activity with a known compound that's released as an active metabolite, and then the other compound being neuroprotective and also being an active metabolite. So we think these approaches could be a significant advance for treatment of glaucoma and neuroprotection, allowing us to get approval off the IOP lowering, follow those patients, and subsequently get approval for neuroprotection as well. So we're in the early stages, and by next year I hope to be able to share more data on these compounds with you. So our overall pipeline, we're currently very focused on moving the first program ahead as quickly as possible to do, again, once every 4 to 6 month dosing for an anti-VEGF and anti-PDGF single agent. And then the next program right after that would be the glaucoma program that we would start in 2017. We've also got excellent data, I think you saw last year, on corneal graft rejection. And as I mentioned, we're working in partnership with ARI on their compounds to deliver something at a minimum every 4 months for their compounds and their therapeutic approaches. And that's been an excellent relationship, and I look forward to hearing ARI presentation later today. So with that, I'll close, and thank you for your attention.